Really hope that you enjoyed yesterday's welcome dinner. Well, I did. I hope you did so too. And I must commend you. All of you really looked stunning. You dressed with your traditional dresses and therefore showed the beauty of your cultural backgrounds. Many thanks for that. Did you take a picture with our famous Golden Mermaid here in Songkhla yet? Obviously with a umbrella. Before we get started, I would like to make some announcements. When you take your name tag and turn it around to the back side, you see a QR code to a satisfaction form, and the committee is asking you to scan this code and give us your feedback on the organization and the proceeding of this conference. The faculty has prepared a shuttle service today at 5 p.m. from the hotel to the airport, and tomorrow at 10 also to the airport. If you would like to make use of this service, please make sure to register in the form that is right by the registration desk. Today until the coffee break at 10:20. Please make sure to register. To all participants staying in this hotel, I would like to inform you to check out until 12, and your luggage can be deposited at the reception. Some have been asking, but many have been thinking about the conference certificates. And I'm happy to announce that you can download your certificate from today on from the website of IMT-GT. As a brief information to the program beforehand, each keynote will take around 30 minutes, and after that you are able to ask questions. There's a 10-minute Q&A session. Let me now guide you a bit through the program today. So from 9 to 9:40, we will listen to a keynote presented by Professor Dr. Lene Lange. Then from 9:40 to 10:20, Professor Wei Zhang is going to address his keynote. At 10:20 to 10:30, there is a small coffee break. And after this, you are going to break out into your track sessions. At 12, there's lunch, and after lunch, you will continue in your track sessions. At 3 p.m. and 10 minutes, we will meet back in this room, and we'll listen to the very last keynote of this conference, presented by Dr. Mushtaq Ahmed Mehman. And from 4 to 4:40, we will enter into the closing ceremony and also the award announcement. Our first keynote will be distributed by a person that has seen the big sister of our Songkhla Golden Mermaid, the Little Mermaid from Copenhagen. Professor Dr. Lene Lange has come all the way from Denmark to Thailand to Songkhla, and her career is shaped by positions in both the private sector businesses as well as public research institutes. After her PhD, the major part of her career was in private industries such as Novo Nordisk, and Novozymes AS. Then back in academia, from 2008, she had several positions, for example, as Dean for Research and as Professor in Biotechnology at Aalborg University, as Campus Director, and then also as Director of Research. She used to be a professor and research leader at the Technical University of Denmark. And since 2018, she is founder of her own company, Bioeconomy, Research and Advisory. Dear Professor Dr. Lene Lange, we are glad to have you and would now invite you to go up the stage and talk to us about the topic, bioeconomy, locally, regionally, and globally. Thank you.
Thank you so much for this wonderful presentation, and thank you so much for the invitation to have the opportunity to talk today. And thank you for a wonderful evening welcoming dinner yesterday, for all the entertainment, all of the dances, all of your costume, everything. It was fantastic. I'm truly impressed. Thailand, I have been coming back and back and back 20, 30 times to Thailand, and you never let me down. I'm never, never disappointed. Thank you so much. I would like to talk. I don't need to repeat my title. I have so many things I want to share, so I will speak a little bit fast. I'll try to make it clear, even though. First, I want to say, I mean the title here very seriously. We forget all of the biodiversity, all of the areas of not using biological resources well. We throw away almost half of all what is produced, 34% just in food waste, and 5 to 10 to 15% of all the residues, all of the industrial side streams we don't use to its full potential. We throw away so much, and to use that better has an impact on climate change, because every ton produced is having lower CO2 imprint, and if you are using it properly instead of rottening half of it. So that is a very important part. It's also biodiversity, more room to biodiversity, more for food security, because of all what we now throw away of side stream and residues, which with modern technology could be used for something more food and even more healthy food. All this I will talk about, and I will just about terms. The BCG you are using here, we never use in Europe. We call it CBE, Circular Bio-Based Economy. So if I write CBE, it's the same as you bio, circular, and green, yeah? So forget, we have to understand each other. So much for this. The first part, let us, Anna, how are you? It's the wrong version of my talk. It's the one which has not been updated from yesterday. I'm sorry, but otherwise I'll just continue. No, this is too much. Time is too precious. So forgive me, there are some mistakes, and they were corrected. Tom, we just continue, no problem. Yeah? I will make sure that the good one will be circulated for you. Okay, the next, Anna, we'll just continue. Sorry? It's the wrong version. It is the old non-corrected version. But I can manage, but it's just such a pity because you can see this is the extra slide. Yeah. Sorry, I have to, don't eat away my time for my talk. I have so much I want to share. I hope I have your full attention. Don't think about the technical problem. Focus on what I'm saying now. Huge opportunity that we are wasting so much is an opportunity. We can do better. We have the technologies. Upgraded use of food, 34% is wasted. Valorizing food processing side stream, another 6 to 12% is wasted. More than 40% lost. Improved use of the crop residues is an opportunity. Sustainable higher value use of the blue biomass cut-offs from fish processing and seaweed, high opportunity. Upcycling of residual microbial biomass, bacteria and fungi, very precious biomass. Making value from organic content of sludge and waste is possible. Upgraded use of residues from non-food processing, fiber crops, etc., a lot of opportunities. We in Europe invested 3.7 billion euros into making the bio-based technologies. This is in a program, BBI joint undertaking achievement, and summarized in a paper where I was the first author, but along with the whole scientific committee where we were advising EU committee, the EU Commission. So I really draw your attention to this. If you in one glimpse, one paper will try to see what is the status of the BCG or the CBE technologies in Europe. This is a paper to consult. Let me exemplify. Valorizing the yellow biomass, that's straw and wood chips. Several different value chains. You don't need just to plow it down or burn it. Please don't let us have so much straw being burned all over the world. It is CO2, it's air pollution for people getting sick of it, and it's smog, but it's also lost value. You can make a sugar platform by enzyme processing where you use the cellulose. You can make a gut health promoting feed from the hemicellulose. And from lignin, you can make a broad spectrum of binders, materials, and other things. And the green biorefinery, that is the grass and leaves and others. Green manure, you can get much more out of such type of material. Protein-rich animal feed, soluble feed protein. And you can make more than just taking a screw press and get the protein and precipitate it. If you treat the pulp left with proteases, you will actually get almost 40% more. Rubisco protein from the chlorophyll. Then we also can make prebiotic feed ingredients from hemicellulose for pigs and chicken, and fish, and also food. We can health and reduce antibiotic microbial resistance. Some of these materials you can make even from the green leaves can be used to sow good feed, for instance for pigs, that we get much more healthy stomachs of the pigs, so we don't need so much antibiotics. And that is so important. The blue biorefinery. We have amazing results where you can have seaweed components. I have listed them all here. Seaweed has components which are so precious for health, for wound healing, for better joints, and even for improved eyes, the sight of aging eyes, MDA I think it's called. It can also be improved by fucoidan. So many health promoting, also skin care and cosmetics, wound healing. So precious. The seaweed is food and delicious, but it can also be made as a biorefinery feedstock and a lot of interesting materials. And there are so much alternative climate-friendly local protein, reducing need for import of proteins if you use them. And I can say it's from seaweed, it is from sea starch, sea cucumber, it's from insects you can grow on your side stream, it is from mushrooms, and you are experts in this part of the world to do that, and of course also from the green grass. Then with all these residues, with all this, I would like to emphasize the following. I have summarized all these different types of biomass and the different types of biorefinery where you can get all products, exploiting the full potential of the different types of biomass. I would like to recommend at this meeting, at the IMT meeting, being just here, for all you specialists, to say this is a huge opportunity for Indonesia, Malaysia, and Thailand to get organized. You can make a strategy which country, which university are specializing in which type of biomass to valorize. And then implement by sharing your knowledge between the countries, regionally, and establishing biomass upgrading at many places in the region. Sharing knowledge, copy-paste experience, growing and do it efficiently. And this way, you can make circular bio-based economy a win-win-win opportunity for all three countries. Try to see it in a systematic way. Not just, oh, I have one feedstock. No, try to see and map all of the opportunity, get the low-hanging fruit where they are most interested in the three countries, separate who has the best knowledge, and get organized and stay in contact so you are building and sharing knowledge. I could end up here, but I have so many more things, because for doing all this here, can you imagine? Just try imagine all these different types of colors of biomass, also the brown biorefinery sludge and the gray, the side stream, etc. All these different types of biological structures. For valorizing those, you need many, many types of enzymes, and you need many, many types of organisms. And you need to be faster in finding the best. And that is why I will spend the rest of my time on describing new methods for you, how you can make even more efficient, function-targeted enzyme discovery. So, but just again, to make it even more concrete for you, what I'm talking about with all these side streams. It is from residues from milling and brewing. It is press pulp after oil extraction from the seeds, sunflower, olive, rape, palm oil. It's from after starch extraction, cassava, whatever you have in sweet potatoes, extraction pulps after sugar cane, beet, sorghum, press pulp, juice, orange, dairy, fantastic side stream from there, microbial biomass, side stream of non-food production, all these areas. So the method, the basic method I would like to share with you is improved enzyme discovery by using peptide-based functional annotation. What is that? Annotation, remember, annotation to Kazime families is just not giving you the annotation to function. And this is a very serious mistake. Many people, oh, I know which family it belongs to, then I know about the enzyme. No, I don't know about the enzyme function. I only know which group it belongs to. Because more of the families have, or most families have many functions. And why am I so much focused on function? Because if you only know the family, you only know the type of enzyme and not the function, you cannot use it to find the right enzyme for your need. You cannot use it to understand the role of the enzyme if you make basic science. So what did we do? Together, Peter Busk and myself, and later Christian Barrett and myself, we made a totally new understanding of how you can make the functional annotation. We start with all these different analyses, but basically, how do you get the peptides? How can you see all the peptides you can get from a protein? If you move just one amino acid at a time, then you get a very long row of possible peptides. Then you cluster, make a clustering analysis of all available protein and find out which share the peptide pattern. Conserved in evolution, it's sharing peptide pattern. If you share peptide pattern in such a clustering, then the next part is you can predict the function. This shared peptide pattern, like it is where it is attaching the substrate, its other essential parts, those is actually the other necessary and the shortcut to understanding the predicting the function. So then we started, after this idea, we started to try to make a annotation and validate if it worked. And then our point here, I think, is too small for you to see the circle, but I'll describe it in a shorter one here. So, cup validation. In the center, we have all subfamilies of glycohydrolase 30. And then that is the colorful rings. And that with all the subfamilies of the same GH30. And then around the whole circle, I have a dendrogram of the cluster of all members of GH30 family. And then if we look at this in a larger, you can here see it. We have, follow me here. Now we have the dendrogram of the clustered peptides. So each of the cup groups is group 19, group 20, 21, 23, 24, and so on. And here you can, for instance, if you focus on, excuse me, if you focus on the cup group 21, you can see that there is in blue, there is an indication of which EC number it has. Similarly, for cup group number 19, we have an EC number. And how come we have an EC number? Because cup group is reflected into having the same, everyone belonging to the same cup group has the same function, and that means that if just one of the family of the cup group is actually characterized, we know all of them. So, improvements, the first one, I think I skipped this one because this is referring back to the first patent we had on the peptide pattern recognition. So in this way, we validated that we were actually able to use the cup, this functional annotation, and maybe I should go back here. Yeah. The three, you can see the three different references I have given below, and don't need to take pictures, I'll share the whole presentation. But the last paper I can see, I have said, try it, it's online. And you see, it was complicated to describe what cup is, but it's easy to use. You simply load in your sequence, and you get an analysis of what are the type of enzymes you have, annotated to family, subfamily, and cup group with predicted function. So now we have a shortcut. That's good, thank you. Yeah, I think. So now we know how we can actually get the predicted function. Then the next part, just imagine that you have not just a one protein, but you like to look at the proteins of the entire secretome of a fungus or a bacteria. And there, it is difficult because most people say, oh, we have a list of enzymes, we have a list of functions, but that's not how evolution works. The point is that you need to have evolution favors the organism who has the right enzymes to mobilize a substrate with the right function. But function is not enough. It's also the type of protein, because the type of protein it's in is determining its pH stability, its different types of the size, three-dimensional structure, etc. So we, or actually I, made the invention that we should combine function and family, and that is into one observation, because that would enable us to actually make a full cup functional annotation of a full organism to functional family profile of a species. And if you then have many species with a different type of annotation, you can make a binary comparison. How many are they sharing? So the ones sharing many function families, observations, are closely related. So in the first hand, we tried both Jacquard and Euler observation. That's two mathematical ways of doing it, where we made an observation matrix and converted it into a distance matrix. This is a bit complicated mathematics, but it's necessary to use it to convert from your function family observation to actually understand relatedness between cyclotomes. So then we are using, and just try to imagine, this was a fantastic moment when we realized, when we took this function family observation as an annotation of cyclotome and used as a basis for actually comparing all 2,000, more than 2,000 genomes or in this case, we selected the 465 most well-annotated genomes. And then we tried to see what happened. And just by having a annotation to the function family, we got all ascomycete fungi to separate, cluster together, and we got all Basidiomycota to cluster together. This means that this makes sense, that evolution who brought and distinguished between Basidiomycete and ascomycete also made a evolutionary pressure selecting the ones who are similar in their cyclotome profile. And can you already now see, oh, now we start to have a guidance of where to look. Because now if we find some, we know for this we want to go there, and so on. The next part, I would like just to say, I hope you follow here, and then I want to go next part. We made a hypothesis, empowered by that we saw it separates ascomycetes from basidiomycetes. And we also saw that Aspergillus and Penicillium, the two big mold genera. We said the hypothesis is that the digestive cyclotome is coinciding with organismal phylogenetic relationship. And that was a very, very bold hypothesis. But actually, when we tested it, and I remember I was attending a conference in Costa Rica when we were getting this result, and Barrett, my young genius bioinformaticist, told me, it doesn't confirm our hypothesis. It doesn't make sense. And that was a bad moment. But then I got an idea. Maybe we should not just, as Jacquard does, count and cluster with relating to what they share. Maybe it's just as important to take into account what they share not having, the observations they don't have. And when we did that, it matched perfectly for all species of Aspergillus and Penicillium. We could make, based on what composition of the digestive cyclotome of its species, we could make a phylogenetic tree which matched the the phylogenetic tree of the organisms. And I have that, and it's complicated, but in the middle, there is a dendrogram of the different types of the enzyme profile relatedness of Penicillium and Aspergillus. Then we have the numbers of the sequences we used. And in the outer next ring, we have all the name of the species. And then we have all the relatedness to the different substrates they grow on. And if you look at all this, all of the clades in the middle dendrogram actually reflect directly into the outermost ring, which is the sections of Aspergillus and Penicillium species. And that has never been proven before, that it is actually a possibility, or this is telling us that digestive profile evolution is an integrated part of speciation. It is evolutionary. It's not just genotype. It is genotype and phenotype is here important for the evolution and for the speciation. And then the next part, I think, is obvious for some of you who start imagining. When we could analyze which one, the cyclotome composition between species across all fungi, we can also do it differently. We can sum up how many observations of different digestive enzyme being cellulases, silonases, pectinases, lignolases, etc. And there we can then try to sum up how many observations they have. Because how many observations they have is an indication of how rich it is in its capacity to degrade, which is a guidance for which organisms are interesting to look into, because it's very rich for enzyme discovery. And in this sense, there are 2 points. The number of observations, unique richness in function, total degrading capacity is a total number. But then you can also say, oh, but many of them have in high number. The redundancy is, if you don't count redundancy, then you have a richness in specificity, diversity. So if you want to find the ones who have the most highest number of different types of enzymes, then those are the species you should go for. And then we analyzed all of these 2,000 genomes available, a little more of genome sequence fungi. And then we found out the hotspot, which are the strongest enzyme producers and which are the richest in different functions. And the result was astonishing, because it was not the fungi used in industry for making enzyme blends. It was not the ones which we are most used to, trichoderma and others, to see that they are the richest. No, it was many surprises. For instance, endophytes, fungi living into plants, they had one of the highest numbers of different enzymes. And why is that? They need to go through all parts of a plant, where both cell walls and other parts and fruits and so on. So they are meeting many types of different substrates. But then also surprise, a stone fungus among the highest. A stone fungus, a fungus living on a stone and almost not growing because it waits till a little tiny part of biomass, organic material, is landing on the stone. And then why is that? I think it's because it cannot be picky. It must have enzyme for whatever lands on your stone. You need to be able to degrade it and actually make growth and reproduce based on it. And all of that is also the reference is giving here. But let me tell here, the top sporing in pectin degrading capacity. There's a small mycena, a small cap fungus, and then Colotrochicum and Aspergillus, etc. But let me just exemplify with a small cap fungus, a small mushroom. That Mycena citricola is a very serious coffee pathogen, and you can see here, I can see why it is so strong and how it is so strong. Can you see it's not making a spot on a leaf? It's making a hole. It degrades totally. And when it grows on the coffee fruit, it is not just making a spot of fungal attack. It kind of sucks in the whole. You can see there's a flattening part where the pectin and so on is totally disintegrated. So you can, when you got surprised that they were so strong, you can actually see it. We didn't see this before, but this is a guidance of how to find the very strong enzyme producers. Then I want to just mention, if some of you have found that we have already made another, instead of COP, we call it peptide pattern recognition, and that is improved. But this is maybe not most important, so I'll skip. You can, I think some of you have already. Now I don't want to go to the next slide because it has a format problem, but we will forget about it, okay? Here are I would like to draw attention to fungal biotechnology on the overview chapter of unlocking full potential of fungi can do. I have now given 3 new methods how you find the enzymes. Now I want to make the overall. Fungi is so important for your BCG, but also for the as for the CBE. And then many of you were interested in seaweed and the blue biomass. We have, along with many good Nordic people, Iceland and Norway and Denmark, authors made an opportunity for seaweed biorefinery, and that's also published. And then I had a QR code for the blue bioeconomy outreach folder, which I really would like to share with you. It will be in the circulated after the conference version of my talk, where you have a QR code and you can find it. I should have brought a lot of them in hard copy, but I didn't. But it's important because the blue biomass is so fantastic, and it's so important, and there's so much potential that I think we should make outreach material so more people understand. And I made the basics of the bio-based society, one folder, and another folder here on the blue biomass. And those two outreach folders, I really recommend that you try and use, because I have met politicians who told me, it was only when I got your folder, Lene, that I understood what you have been talking about for this time, about so important of enzyme and upgrading waste, etc. And the similar for people's interest, it is now translated, the basics of bio-based society in 8 different languages. And I didn't advocate for it. It was just people, can we make a new and can we make a new? And the other one for the blue biomass is coming now in 5 languages just in a short time. So I really recommend, because more people need to understand the importance of using the bioresources better. And we are speaking and not often get understood. And therefore it's important that we are able to distribute this knowledge. The new bioeconomy opportunities, I have a few more minutes. I would like to mention that the microbiome research, follow it closely. Microbiome research has made paradigm shift of understanding microbiome is the missing link between food, nutrition, and health. Everyone should be aware of that this is important, and everyone who is interested in biorefinery, making better use of the resources, we can then, when we know the microbiome, for instance of the gut, we can make high-value, health-promoting products out of all types of biomasses, especially plant cell wall-rich biomasses. And we can use bacteria to ferment such biomasses, and so we can make both prebiotic, by enzyme treatment of your biomass from the plant cell wall polymers. We can make fermented by, for instance, lactic acid bacteria or fungi to make probiotics, and the fermentation make anti-inflammatory compounds, so we can improve gut health. It's doable, we have proven it, and it's fantastic. There are a lot of new papers out, and I'm privileged to be part of many of the new reviews. Microbiome research needs higher priority. I have said here, microbiome, so many, all the blue here, food security, drought tolerance of plants, soil fertility, public health, stopping biodiversity, antibiotic resistance and spread. Being prepared for new pandemics, we can look at the wastewater treatment plants and identify here in the microbiome the rising of new pathogens, but all antibiotic resistance genes. This is serious stuff. I really hope you will take this one. It is so much needed that improved and proactive local and global knowledge sharing about microbiome. I am going so far to say that this is not just interesting science, this is an ethical obligation to share, because it's so, I just listed all the parameters where microbiome is important. We are losing the diversity of microbiome in a modern society, not just biodiversity in the animals and insects and so on, plants, but we're losing microbes in our microbiomes. That is making soil and plants and everything more vulnerable and less productive. And for our gut channel, is lifestyle diseases has much to do with this. Also diabetics, a lot of diabetics in this part of the world, it's important. So we made, again, I took the initiative to make a paper here on microbiome ethics, and you can study, I tried to summarize it in one paper or one figure here, and it is, I mean, microbiome ethics for One Health by the analysis: do good, don't harm, respect, and act justly. Those 4 parameters is the way I made the ethical analysis, and I think it sets standards. And I want to make sure to emphasize that in the paper we also describe and recommend that we should make, as we have the international treaty where there is the 64 of the food crops of the world is by UN declared common global heritage. We should also say that the gut and the microbiome, those organisms should also be part of common global heritage, because it's not owned by anyone. It's part of us being able to eat, because it's without the guts, the microbes in the gut, we don't get anything out of the food. So I see it as a continuum of the international treaty. So, importance of microbiome health for public health, I think I have mentioned, and I also think I have mentioned, and I would just want to say, improved use of global bioresources can impact climate, public health, biodiversity, local and global jobs, and I then add business. Business is, there's so much good business in using these 40% we are now throwing away. And there is so much good business bringing new knowledge out to people and make value from all your knowledge. You should also use that to make, it should reach the end user, and it can only reach the end user by that you also get it sold in products and making it available and accessible. And making new gut-healthy products can also be not just accessible and available locally, but it can also be affordable because your starting material is cheap. So with this, I would like to finish my talk and a closing remark. Circular and bio-based food, residues and side stream represent a huge opportunity. And I really, really hope you will use your wonderful collaboration between your 3 countries and expand your platform you can build to even more countries in the region. And then I also hope that you will try to study, difficult for me to really describe for all of you, the 3 new methods where you can guide your enzyme function targeted enzyme discovery by using these 3 methods, so that we can valorize such a huge amount of biomass which is now going wasted or downgraded. So with this, I would thank you so much for your kind attention. Thank you.
And we thank you very much. I would like to announce that the keynote presentations will be ready for download after lunch, so you will not miss out on any updated information from the presentations. I would also invite all the guests sitting right back over there to feel free to come a bit more into the front so you can read the presentations of the presenters better too. Now I would like to open the Q&A session to the talk of Professor Dr. Lene Lange. Are there any questions from the audience? Please, yes. Professor Singh.
I'm excited to hear what you want to ask.
Thank you, Prof. Lange, for a very inspiring talk. I was at the edge of my seat, almost fell off. But anyway, I have been, coincidentally, I've been working with valorization of bioresources from all these biorefinery that you have color-coded them.
Could you take off just the mask? I have difficulty understanding.
Sorry.
Just 1 minute.
Better now? Okay, so my team have been working on the biorefinery that you have color-coded them. I'm working on the red, blue, green, and gray probably, so which is interesting. But I always find it a challenge to translate our work into a real-world application, where we did have very good results from the laboratory, But to have it upscale and to have it being solely or being transferred to an industry partner, that is always a very big challenge for us. Because in the university, we don't have the capacity. We couldn't produce in kilograms or in tons where the industry needs. We only produce in grams. So could you share with us any of your success story to upscale your products or maybe to go into technology readiness level of 9 and so on that you have experienced in your work? Thank you.
I have two levels of answer. First of all, try to familiarize yourself with what is the state of the art in the type of feedstock you're working on. Because there is so much knowledge out there. Don't start from scratch. So if you cannot upscale, sometimes it's because it was not the right way to go, the process. Other people tried and it didn't work. So that's one part. The other part is find out if you have the right process in the sense of enzymes, microbes, because sometimes you can, is it robust? Does it work? And so on. The next part is your feed. When you upscale, every biological process, upscaling is a hurdle. It's an obstacle. And I'm not going to say, oh, you just do so and so. You have to take serious that upscaling a biological process is a research task in itself. And don't say, oh, no, I'm ready for upscale, so I don't need the science anymore. No, that's not how it is. You have, I have 20 years with enzyme discovery and upscaling with Novozymes, so we really, really know. We need to have the people who understand, the researchers, along the whole upscaling, and then you need to team up with the one who knows about the engineering of larger volumes, your feedstock handling, your pre-processing needed or not. Yeah? So it's a serious task, and it's a hurdle, but start by looking into literature there and take the paper maybe I have made here about what we have done with all these shoots program in Europe, and then get inspired by that, and then follow all the real papers on the individual. So we need to share and use the knowledge. Biology has more knowledge in the public domain than any other field. Use it. Share it.
Thank you.
Thank you for the Q&A session. I'm sorry, I have to close it. We have to continue with the session.
Ask me in the break.
Feel free so, please. All right, now I would like to, please, Professor Lena, stay on the stage. I'm just inviting Associate Professor Dr. Anjana Pratep to show our appreciation and present you with the token of appreciation. To continue with the session, another invited keynote speaker has also come from quite far, from Australia to be exact, is Professor Wei Zhang. He has postdoctoral experience at the University of Tokyo, University of Adelaide, and University of Cambridge, and his career is shaped by his passion for marine bioscience. He's known to be a visionary leader that recognized the economic and social value of Australia's unique yet underexplored biomarine resources since 2000. Professor Wei Zhang is a globally recognized leader in research and teaching experience in industrial and marine biotechnology. As in 2009, he founded the Center for Marine Bioproducts Development to pioneer translational research for the establishment of a new marine bioindustry in South Australia. Currently, he is Research Director in the Marine Bioproducts Cooperative Research Center, inaugural President of Australia-New Zealand Marine Biotechnology Society, and he is the chair of Marine Biotechnology Subcommittee of Australia's National Marine Science Committee. We are happy that you are here, and we are excited to listen to your talk now on the topic of circular blue bioeconomy by design, integration of marine biorefinery, and advanced clean technologies. Please.
Thank you, Tom, for your introduction. Good morning, everyone. And first of all, I would like to thank the conference organizer, PSU, for your kind invitation to come to Songkla. This is my first time to this area, but the party or dinner yesterday show something different from the other part of Thailand, also something in common, but definitely enjoy the evening yesterday and also have lots of fun. Hope you all had the same experience if you've been first time to this region. So I will start my talk, just acknowledge what Leila just talked about, this different corner of the bioeconomy. I will talk about today is about a blue bioeconomy. And by corner, the blue means aquatic, very much so. I focus on ocean territory here. And as we understand, the Earth being covered 71% by ocean, and it can go as deep as more than 10,000 meters. So what does it mean? That means they really have very unique resources there for our bioeconomy. And this particular talk about is biodiversity or bioresources in the ocean. However, I think this would be a great opportunity because we have been working quite limited on the blue bioresources. So this is where I'm thinking for the circular bioeconomy. So we have a wonderful opportunity. We can start from a beginning where we can design the blue bioeconomy, which is, of course, can be circular or is circular. So that's a whole thing. But of course, for today's talk, given the time limitation, I've only talked about one part of technology solution with the integration of the marine biorefinery, and particularly have been driven by the advanced clean technologies to achieve our sustainability goal. I will start first maybe introducing a little bit where I come from and also why this is relevant in Australia, New Zealand context, and of course in your IGT region and also globally. Start with this one. Australia actually is the third largest, what we call the EEZ, exclusive economic zone, which is basically 200 miles away from the coastline. That is where we can name as the country's ocean territories. So we have the third largest EEZ zone, which is about 8.6 million square kilometers. Australian land size is about 7.6 million square kilometers. So basically it's larger than our land territory, if you think. And also, if you look at New Zealand, the same thing, New Zealand is ranked number 9, the EEZ zone in the world, against 4.1 million square kilometers. It's not small. Okay. Combined together of Australia and New Zealand, so we are ranked the world number 1, sort of marine territory. Is about 10 or 12% of the global EEZ zone. Global is about 101.7 million square kilometers. If you know, how big is France, number 1, and the US is about the same, is about 11.7 or 6 million square kilometers. So that's to give you some understanding. However, I have a question asking you. This is IGT region. So how big is your ocean territories here? Anyone can give me some indication? Maybe I will quickly say Indonesia is ranked number 6 in the world, is about 6.3 million square kilometers. And both Thailand is ranking 64, is about 0.3 million, 300,000, so close to 300,000 square meters, square kilometers. And also the Malaysia is about 334, is ranked 30 something, So you combine together, is ranked number 5, just close to UK. So again, I just want to demonstrate that there's a huge opportunity, not only for Australia and New Zealand, also for IGT region. And for this reason, as in this conference or your platform in terms of UNINET network is very useful for you to working on the circular bioeconomy based on land territory, resources, and very importantly, you would build a very significant opportunity for your 3 countries in the marine territory. So that is a key point I want to make. Next part, I'm going to introduce in my current organization, which is called Marine Bioproducts Cooperative Research Center. So this is relevant for today's talk of bioeconomy. As I said, this is the first Australian national industry-driven or research flagship for marine bioproducts, where we try to build the 3rd generation of Australia's marine bio industry. When I talk about 3rd generation, you'll be wondering what is the 1st and 2nd generation. The 1st generation of marine bio industry is what we call the fisheries. You basically go to catch, where the ocean provides for you, the gold provides for you, right? The 2nd generation is aquaculture. We have more control. We can grow the stuff controlled. But both the 1st and 2nd generation, they're producing the seafood, mainly primarily for seafood for human consumption, right? Yeah, the 3rd generation is industry based on the marine bioresources other than seafood. That means not just only seafood. Seafood still can be considered, but it's not just a seafood. That means marine drugs and also the enzymes, marine biomaterials, agrochemicals, and also health, nutrition, skincare. So you can think of any of other kind of products which can be derived from both from terrestrial biomass, and of course, they also can come from the marine biomass. So this is where the center was being funded by the Australian government, and we officially start from July 2021, just about 1 year old. And as I said, this is really to use the innovation, R&D innovation to drive the Australian 3rd generation marine bio industry. So this is an organization formed based on the co-investment from the Australian federal government and also the 11 university research partners across the whole country in Australia, and also about 6 industry partners come from mostly from different states of Australia and also from international space. So those all together have secured 10 years funding as starting point, $270 million last year, and to really developing the innovative technology solutions and the product solutions for the industry. So I would try to emphasize in the way we set up the research center is based on the supply and the value chain. So basically, we're not just only develop a piece of technology, we're developing technology solutions and where we can drive the supply chain from the marine biomass production and to the advanced manufacturing to process the marine biomass into functional ingredient, and then of course move into the Australian marine bioproducts innovation, where of course you can develop all these different new products and validate these products, use the science where they can go into not just only Australian market, but aiming for the global market access. So therefore, our partnership across both industry, research, and the government, also you can see the distribution there. We have the partner across all the supply and value chain, where we feel that by working together in collaboration, we would be able to build a new industry. And our mission is very simply, so what we try to do is make Australia a global player. So in the marine bio industry, which we can contribute at least $1.5 billion per annum and 10,000 new jobs per year by 10 years time frame. That's just a starting point. I said the potential for Australia is just more than that, can be 100,000 times more than that. But at least we have to get this started. So this is where we have set it, this particular research center funded. And our research program, the similar way we've been developing our research program is across the 3, the supply chain, where our program 1 is called Sustainable Marine Bioresources. This is where how we can grow the marine bio products containing biomass. The second program is called Innovative Bioprocessing Technology, which we can make in marine bioproducts. Of course, this including all the cleaning technology, including what Nina just said, the enzyme is very important, the microbial process is very important, enabling technologies for that. And of course, the third program is called Australian Marine Bioproduct. It delivers product to the marketplace. What is important components of, we don't call a program, but in fact, we call it Connect, Educate, and Train, which called CET. This part of program is really to grow the next generation of our industry and scientific leaders, and also train the workforce, which is being embedded with the 3 research program is underpin all the development of technology. We also see it's quite important for human talents, for us to be able to translate the technology innovation and into the reality. So you need the people to do this stuff, right? So that is we're emphasizing on what we do. So that's possible to give you a good idea so where I come from. So now I'll come to the topics today. I still want to emphasize in the blue. I talk about the blue bioeconomy. We understand that the ocean, I think, have more biodiversity. 30% of Phantom exclusive marine, but there is only one, exclusively terrestrial. So it's something to give you an understanding. There's huge unrealized, untapped opportunity in our ocean. That is definitely. And another thing, if also we've been concerned on what ocean can offer to us. Another way, we must do this by design and be sustainable. I have to say that the same thing, our marine species is also under significant risk. So basically, the estimation about more than half of our marine species may disappear, in about another 8 years. Okay, so that is a huge risk. Therefore, when we talk about our explore the marine bioresources for industry, very much so, we have to understand how would manage this sustainably. Okay, that is very, very important. But reality is 60% of our human population, which heavily rely on this marine, resources, and now have been, degraded and also may not be sustainable. So this comes into the topic. I think I don't need to speak too much here because yesterday this company CEO talk and also Cornelia talk is already leading to the circular economy. What I want to emphasize here, the circular economy, is good thing is one about the problem we created, how we solve it. Another thing, actually, can we prevent the problem in the first place? Let's make it really sustainable or circular. So therefore, as you can see, the circular economy is very much about everything is going to be low waste or low environmental pollution, because this can be 100%. That is ideally, to be always in the circular context. But what I think is important for, I just can say this one, I guess the question we've been under pressure because linear consumption is not working. We want to do circular. But circular, I think, is not about solve the problem we have today. It is really huge opportunity by design. I was thinking this even more so for the developing world, which we can start from beginning because our economy not will develop as developing world. Developing world have existing mature industry based on fossil fuels. They have more problem to manage, how they can transform their existing economy to a circular economy. However, for the developing world, I think you have a huge opportunity to build this by design and also by deliver. So this is where I put this called CBBD. And the CBBD, I think I started with sustainability index, okay? Not starting, can we develop a technology which is circular? I think you have to ask yourself how we define the sustainability. For this moment, I think it's more maybe coming, and it's 4 bottom lines. Economics, I think unfortunately the world has been driven by that way. We want to make more money. That's can become in their economic viability. Social sustainability, environmental, and also ethical. I think NELA has done very good work on the ethical stuff, in terms of the bioeconomy. I think these 4 bottom lines for this time being, and they have to be on the top of everything in terms of our science discovery, not be so, but particularly for us in terms of technology, innovation, and development, and also business innovation. So that's would it be your guiding, principle or baseline, and to think about for any of your technology innovation and business innovation. However, this is mutual interaction, okay? But you have to start with sustainability definition for your business, for your technology development, and therefore this can be driven, this circular economy, in every step in terms of how you are going to make and use, reuse, remake, and recycle. And all this have to be guided for each of the steps of your technology and business development using, I said, at least now, there are 3 tools you get applied. This is life cycle assessment, tech economic assessment, social and ethical assessment. So what does this mean? That means for any of the technology, most of our scientists work on one part of the bigger solutions, right? But when you do that, ask yourself where your technology innovation fit into sustainability. And of course, you get to be working with those experts if you don't have expertise in LCA or TCA or SEA, and working with them, and therefore you would be able to have very good assessment right at the beginning. So this is my technology innovation can lead into sustainability, or I'm driving by the sustainability goal. So therefore you know that your technology will work, will be able to translate it into the industry practice because you have addressed of all the sustainability principle. Without that, of course, there's no way you would be able to, to be used to be, successful scale up in the future. And this is something I will try to spend a little more time here. This is what I mean by design, and it is really to be able to start with the bottom lines and then move up in terms of assessment. But unfortunately, most of us as a scientists or technology innovators, and we are only working on a very small part of the bigger solution. Sometimes we don't see the big picture. And we don't also have expertise to work on this one to be able to develop something which think is really useful and valuable. So therefore, the collaboration. Yesterday, I think your former minister have emphasized on the collaboration. I actually take that very seriously as a key message for everyone working in your small field. You're thinking a little bit bigger, outside the box, to be open-minded, and also try to identify your good collaborators. Those collaborators can help you save lots of time, because sometimes you can waste your whole life to work on something which is not meaningful, although we think it's meaningful, but in reality it may not be meaningful. So it's quite important. And also, of course, for the younger generation, I think that's even more important. Change your mindset in the way you learn, you work, and also interact with other people, and to see how we would be able to collaboratively, develop a bigger solution to solve the problem. And so therefore, I would give you a few case studies from what we have done. I don't pretend that we have a solution, everything in there, but I've taken 2 important technology development where I think we can create some of the circular blue bioeconomy solutions. The first, the one I'm talking about today is marine biorefinery. So biorefinery, I think now everybody understands it. Yesterday, one of the Indonesia Victor has provided a good example with this Pancari oil development. At least that's one good example. But I was asking yesterday, so the oil comes from the leaves or whole plants, but after extraction, the 3% of oil, you have many of other waste products. How would you going to use it? So biorefinery is basically an integrated process, it's a cascading process. You can use a variety of different technology. However, I talk about clean technologies, and to produce multiple products by comprehensive use, preferably 100% use to achieve a circular economy of a single marine biomass and with minimal or zero waste. I talk minimal because ideally zero waste, 100% use towards the circular economy. So ideally is 100% utilize your resources and also without any waste. But clean technology means that you get to have minimize, I talk about minimize, possibly is the better way to use it, your water use, your energy use, your resources use, and chemical use, of course, so to avoid any sort of secondary and tertiary contamination. The first case study comes from our own work here. And I don't need to introduce too much because seaweed was the example, also case study, and Nila has talked about it. This is when I start from the beginning. It's macroalgae seaweed biorefinery. We talk about brown seaweed. First, I start the economic analysis in a way. This part was too small for you, sit in the bank to look at it. But I said typically your 3 tons of the seaweed you can I talk about fresh, and typically with 3 tons of seaweed, and you possibly can produce typically if you're producing your arginate, for example, brown seaweed is about 1 ton, so it's about 30% typically what you can get out of it. What I'm saying here, I don't want to go into much details here. If you do a single product, use the seaweed either as a food or just a producing of arginate, or some of the polysaccharide is a polysaccharide. So what end up there is you possibly can generate a product total value, even the waste can be used for fertilizer or feed. You generate about $25,000. That's just what we estimate in the current industry. And your 3 tons of seaweed raw material maybe about $5,000 per 3 tons. That's a starting point. It's good business. You do make a net profit, about 10%, minus all your processing cost, any of other stuff. If you do biorefinery, by comprehensive utilize of the seaweed, you can produce, of course, the seaweed polyphenol, fucoidan, and arginate, and also high quality food grade, a different grade of arginate, of course, and it also can produce nuclear fertilizers. And all this actually can create a value about $160,000 instead of $25,000. That's about 6 times higher. However, your net profit can be more than 200%. It's about 10-fold increase. So again, I want to demonstrate here to do a circular bioeconomy is not just your imagination. You actually will sacrifice on your economic gain. Reality is you will gain much more than what you will normally get. So this I want to address the first economic assessment, right? And of course, underline that I did show something here, and of course, move there. And there's more opportunity for produce high-graded medical application. Of course, your dollar value can be even more fantastic. But this without using much more resources. However, you would be generating much more profit. I can quickly go through here one, which basically this where I think you would be able to start with a biomass, in this case, the macroalgae. You can do the physical and chemical fractionation, clean chemical, preferably water, for example. And of course, you enzyme process, the fermentation microbial process. You can say they are the 2 enabling technology platform, which they can help you to do the chemical and the biochemical transformation of those into these different platform chemicals, and of course can be developed and produced for multiple products across all industry sectors. However, technology side of the story is that when you want to develop these more advanced products, and of course you need advanced processing. So if you like seaweed as example, if you only harvest the seaweed or grow the seaweed, and it can easily turn into aquafade, or you just give it washing, you can give it in salad, you can drying, of course, can be preserved in slacks and all food processing. Of course, go through this process of advanced processing, you're not only actually upgraded your value and produce a diverse range of product, and it's truly that they also would have more functional applications there. I want to emphasize. I give you some simple example to demonstrate here, and this actually work mainly been done by one of my students from Thailand, and that's why I choose this particular case studies. And he's working in Australia on our Southern Australia seaweed. Now he's back to Thailand. He's working on your Thai seaweed. So basically, the dried Echinacea radiata is a brown seaweed, which is the most popular in Southern Australia, or most abundant, put it this way. So basically, the typical process, you do a crude extraction, you generate residue or product, and of course, here it gets more complicated a bit with bit residue, and your frotani enriched extraction, you get enzyme process, you get another extraction. You also can separate low and high molecular weight. I demonstrated with a very simple example. what I want to say here, you potentially produce one single product, now you can produce 3 or 4, 5 different products, okay? What I want to demonstrate, actually, those products do have a function. They're not just for the sake we want to fractionate and produce different components of it. So one area we've been looking at, particularly is with my sitting in the medicine college, we're looking for what can be used for improve our health and function. One area in the gut health is the prebiotics, right? Because apparently is rich in fiber. So where we demonstrated, as you can see from this diagram, and actually they can compare to the control. Of course, in 2 to 3-fold, they're also quite equivalent to the inulin, which is one of the best positive control at the benchmark of commercial products. So basically, this in terms stimulate the short-chain fatty acid production, which is quite important for prebiotics to function. I just want to say. Second one, look at the gut microbiome, and particularly looking for if they can promote the good bacteria and bad bacteria. Again, we demonstrate one of the products with no molecular weight polysaccharide fraction is one of the biorefinery products can increase the beneficial bacteria about 10-fold. And also, very importantly, these different products, of course, have different function. For example, the high molecular weight, polysaccharide doesn't function, so we can identify the specific fraction of the polysaccharide for application. And also, we can identify in both of the products we're producing PFSF can reduce the beta bacteria by about 10-fold. This is called about enterococcus. Okay, just I don't want to go into too much details. The idea I want to demonstrate here, really you can see they do have different functions, therefore they can be developed for different applications. And of course, the polysaccharides also can increase stool bulk. I don't want to go into too much details there without affecting any of other of the organ functions. That means they're safe to use, and also they can, better to control your health. The next one also for some of the metabolites, we talk about there's also protein components in there, which they basically have 2 types. One is phenol P cresol production. Again, what we demonstrated here, they can reduce the whole series even. Of course, our polysaccharides production from there can reduce about 80% of second phenol and also phenol concentration. That is very beneficial. And of course, they can also reduce 95% of the P cresol. Again, is very significant, and also polysaccharides about 60%. So what I demonstrated here, the similar idea. So basically, any fraction you have created, so we have a purpose. The purpose not just only to make a chemical, you make functional chemicals. Their potential can be used as a product. So I only take that one because given time from one demonstration. And we also can demonstrate or by simulation. Not necessarily you have to do your real scale up, which of course it costs $1 million or tens of $1 million. And very importantly, you can use those type of simulation tools. You can simulate, if you scale up this to industry scales, what economics looks like, what the environmental impact looks like. So all this type of analysis can be done in silica rather than in silicon, rather than an experiment or in your factory building. So we have done this. I just want to show this different scenario you can make a design. Not necessarily just one way you can develop a process. You can really make this design. So what we have want to show here is by doing this biorefinery, we can actually improve significant economics in the payback time, net present value, and sometimes if you use different scenarios like scenario 3, you may have this negative net present value. So basically what I'm saying here, without cost to $1 million, tens of $millions, you can do only maybe say $20,000, $10,000 of a project. And if you can find the experts to work with you, you can do this take economic assessment, environment impact assessment. So therefore, you can clearly understand your economics, understand how this biorefinery clean technology can help you, to achieve the full bottom line sustainability. The second study, well, I'm focused on the safely processing waste product is lobster. So in Southern Australia, we have one of the most beautiful lobster industry there, is Southern rock lobster, beautiful. So the lobster process industry, if you process lobster, you leave with shell, head, and liver, typically over 50% being wasted. Okay, so what we have developed here is basically lobster shell, head, and liver as a starting point, and actually the raw material cost maybe per ton is about $3,000. We pay them rather than they have to pay the dollars to landfill. And then we do the processing to produce lobster, because of biochemical decomposition, lobster, any of the shell, head, and liver are the proteins, oil, minerals, and the polysaccharide. So what we did is we can actually develop the process to produce lobster protein peptides, functional, lobster chitin chitosan, lobster calcium, and lobster oil. All this can be applied into many different product development where we can turn our $300,000 per ton raw material into something over $100,000 per ton in terms of products value we've been doing. And I already talked about 56, 70%. Also, very interestingly, also need to talk about the safety processing waste is a huge resources. It's any landed, from the ocean, and about 50, 70% wasted. Okay, so lobster just one example to demonstrate this one, and of course they pay the money for landfill or have to be given. This is where we deal with the product. So the first critical part is for the analytical, characterization of the composition. This we characterize the shells, the head, liver. We understand what they are. If you look at them, they are all very useful rather than you discard it, right? Yeah. So I just emphasize what we do. So the process we start with is first with oil extraction with cleaning technology. Of course, sounds expensive, but supercritical carbon dioxide. But the oil is very expensive. I can demonstrate to you. And we can use this process to produce lobster oil. This lobster oil, with this cleaning process we develop, and actually they are rich in omega oil. They are also lower in the heavy metal. Maybe next one, I just want to show quickly, don't want to spend too much time, a toxic chemical. So this is understandable because those will be stay with raw material rather than being extracted by the supercritical oil extraction. So what this has been done is the industry have used this oil, lobster lipid. This is what the stuff looks like here, and you can make into olive oil in 10% or 1% up to 1 to 10%. This is olive oil. It's a flavoring oil. The olive oil you may sell $5 per liter, and this lobster flavored oil $20 per 200 ml or 100 ml. So you can see it makes significant economic gain here. Okay, so this is something I want to say. The biorefinery part, as lobster shell as example, again we try to use energy efficient technology, the microwave assist enzyme sort of digestion. So first step we make of the this sort of deep proteinization where we can make the protein hydrolyzed, use the enzyme. So that's the first step. Again, I don't want to give too much technical details there. So we do produce beautifully, one is a protein powder, and also we can produce a phasal process. Again, we can use very mild process after deep proteinization, use lactic acid again, this is organic acid with for deep pigmentization, and also produce a beautiful chitin. And of course, all the other part is the calcium is going to the salt in lactate salt, calcium. And you can see this actually all been done in a mild, clean, sort of conditions where you don't use any, toxic chemicals. It's water-based organic acid, supercritical carbon dioxide, and also enzyme process. What it does, okay, I just want to show you again to see how useful those products we have produced. One is the lobster, protein hydrolyzed or the protein, the peptides. Again, looking at the nutritional value here, we characterize the nutritional value, and again, you can see they are very useful, as nutritional supplement where we try to develop an aging nutrition, product. Another one is chitin. Chitin, you may say one application we demonstrated here actually have a significant ability to absorb the fat. We talk about here is butter, vegetable oil, canola oil, any sunflower oil. What we demonstrated, this can show an example, and this is a commercial product called fat broker. So when you eat an oily food, you can take this capsule, which can absorb the oil. You basically cannot digest it, it passed away. but you still have very good taste of your food. This is commercial chitosan. This is our lobster chitin. They can absorb 6 times more oil. You can see the olive oil here. This is our lobster protein is 8. This is about maybe 1.6 times. And of course, the vegetable oil again is reaching 12 gram per gram. This is about 1 gram. So it's about basically 6 to 8 times more of oil absorbing capacity. I think it is because of our process producing the chitin have a structure, they can retain or absorb more oil or fat. Okay, so that's its application potential for weight control. Another one, the lobster calcium being produced is bio-derived calcium, and you can see compared to commercial minerals, they have much more solubility and also have much higher bioavailability. This is quite important for us to take a calcium supplement, right? So where I think they've been developed as a dietary supplement. Again, we still use the economic and also the environment impact analysis using our simulation tool. These tools, again, allow you to design your, put your technology integrated into a process where you can, of course, have different design and you can compare their economic viability and also environmental impact here. Again, we demonstrate here. So by doing so, you also give the significant economic gain, not just only you minimize the environmental impact and for this process. So again, so don't want to spend too much time. I really, given the time limitation, I almost reached my time, right? So I'm going to finish up here. I think the take-home message for everybody here. I talk about the circular blue economy by design. This is very much so for any of the circular bioeconomic by design. You can do the similar way. But what I want to emphasize here, the marine bioresources is a huge renewable regenerative feedstock. And they are going to be significant resources or bioresources for our next generation of industry, bioindustry. For your region, IMT, very significant. For us from Australia, New Zealand, I demonstrated, we have a huge untapped opportunity. And the circular economy by design, which of course have enabled by clean technology and biorefinery, and of course the full bottom line sustainability check is what you would like. It's not just only make you more profitable, more sustainable, which is a key for now and for our future. So with that, I would like to finish up by this slide. I'm organizing the 13th Asia Pacific Marine Biotechnology Conference in Adelaide from 2nd to 6th October 2023. And what I want to emphasize here, the key features, we try to balance of academic and industry participants. We realize that collaboration is important to bring all parties together. And we do run an industry investor policy forum, the government agency, and you can participate, business industry can participate. We also run the ECR early care researchers and the forum to encourage the young generation to take actions to working together. We also encourage the women leadership. So we encourage any of the female scientists in this area, you want to develop a leadership, come to participate. We have industry exhibition, so industry can come over again. So with that, I will finish the presentation, and thank you very much for your listening.
And we thank you, Professor Wei Zhang.
Thank you.
I feel sorry that I don't give you the opportunity to ask questions. We really have to continue because after this we are going to break out into the track sessions so that they can start at the allocated time. I would like to tell you that now there's the last opportunity to register for the airport shuttle today at 5 p.m. and tomorrow at 10 a.m. and you will find the form at the registration desk. So make sure to use that service if you would like to. I also announced you to use the satisfaction form. Now all problems are solved and you can enter and access this form. At 10:30, so in just some minutes after you maybe grabbed some coffee and some snacks, we are going to split into the sessions. Professor Dr. Hamam Riza is going to talk about AI for humanity, industry, and environment in Songkla Ballroom 1. In Songkla Ballroom 2, Dr. Anshanok Jung Sombat will talk about design, science, and digital innovation for BCG. In the Phương Pha room, you can listen to Dr. Sudesh Kumar about biohydroxyalconate, biodegradable plastics for a sustainable future, promoting bioeconomy, circular economy, and green economy. And Professor Dr. Sutawat Benjakun in the Beach Lounge will talk about valorization of shrimp processing wastes, shrimp oil, and Shaituligo Sakar. I'm sorry that I pronounced this before you got your token of appreciation. I would like to ask Associate Professor Dr. Anjana Pratep to the stage and address our appreciation for your contribution. I'm sorry. Thank you. I wish you all great benefit from the sessions, and we see each other at 12 for the lunch break. Thank you.